Good morning, ladies and gentlemen. It's nice to be back here in California again this year. My name is David Millett. I'm ExCellThera's Chief Executive Officer. As you can see from our cover slide and tagline, by making better cells, ExCellThera is focused on providing better lives for patients and their families. I'll let you read the legalities of this disclaimer slide and move on. Okay, let's begin. ExCellThera was founded in 2015 and launched its first clinical trial in 2016. So we've been at this for a while. We position ourselves as a world leader in blood stem cell expansion and rejuvenation, and I hope that this presentation will help you understand why. ExCellThera's activities are comprised essentially of three segments. Our UM171 powered enhanced cell expansion and rejuvenation platform designed to help cell and gene therapy biopharmas to deliver a greater number of more functional processed cells to their patients for improved clinical results. Our second segment is UM171 cell therapy, which has been dosed in over 115 now patients with severe blood cancers across the United States, Europe, and Canada. We also have a very exciting pipeline of next-generation cell and gene therapies. So first segment, our enhanced platform. One of the biggest problems in cell and gene therapy right now is the low functional cell dose from process-induced cellular stress and exhaustion. Even if the resulting cells from a cell therapy or cell editing process have been successfully modified, there are so few and the cells are so exhausted at the end of this process that this often leads to poor engraftment and poor clinical outcomes. The solution, our enhanced platform. In 4 words, more cells, better cells. I present you with the enhanced proprietary platform for cell and gene therapy processing. On the left is the starting materials, which can be any source of stem cells. May it be bone marrow, mobilized blood, or cord blood. On the right are the resulting expanded, rejuvenated, and expanded and engineered therapeutic HSCs to treat various indications. In the middle, we have our UM171 powered cell expansion and rejuvenation technology, which is compatible with any gene therapy modality. So let's talk a bit about UM171. UM171 is the small molecule that is considered, I believe now, the standard for ex vivo HSC cell expansion and rejuvenation. It is patent protected worldwide. It has a first-in-class mechanism of action. It's been clinically validated in over 100 patients, 115 to be exact. It's continuously cited in peer-reviewed scientific journals and publications. It's used by various industry and biopharma partners in the sphere of cell and gene therapy. And it is supported by a drug master file, or DMF, as a key reagent for ex vivo use in cell and gene therapy. So as I mentioned, UM171 has a first-in-class mechanism of action. On the left, you can see how, simply put, UM171 degrades a repressor complex called CoREST, which acts as a brake, if you will, on HSC self-renewal. On the right, you can see how this has a profound impact on HSCs by improving the number and the quality of stem and immune cells, leading to a healthier, rejuvenated graft. So as I was mentioning a little earlier, even if cells are successfully modified through a gene therapy modality, may it be CRISPR, base editing, LV, just to name a few, at the end of the process, they are often so few in numbers and so exhausted from the process itself that there's a significant risk of engraftment failure and poor clinical outcomes. By incorporating UM171 in the process, this leads to a greater number of more functional cells, thereby solving for the risk of poor engraftment and poor clinical outcomes. UM171 significantly expands different cell populations, including exponential increases in immunomodulatory cells. These cells populations, as you can see here, include dendritic cells, mast cells, and NK cells, which are linked to better clinical outcomes. Now, this particular slide shows actual UM171 expanded cell therapy doses that were infused in our patients, starting from doses that were so small they did not even meet the minimal criteria in terms of cell dose for clinical use. As you can see on the right, even from these very small starting volumes, our UM171 platform produced so many cells that, unfortunately, we had to discard and trash a number of cells in many cases due to dose capping. On this slide, we set forth a number of actual examples of programs we currently have ongoing with different partners and collaborators in the field of cell and gene therapy. As you can see, UM171 has many use cases across various cell lines and various clinical indications. So we are partnering and collaborating with various institutions and companies in the cell and gene therapy space. Our partners who have actually tried it loved what they saw when they tried it. So if any of you are working with HSC's ex vivo and want to expand and rejuvenate cells with UM171 for better outcomes, please do not hesitate to reach out to us to see how we can provide you access with this game-changing technology. Now on to our second segment, UM171 cell therapy. In terms of overview, UM171 cell therapy is an expanded allogeneic stem cell transplant product to treat blood cancers using cord blood as a starting material. It's received multiple designations from the FDA and the EMA, including RMAT, PRIME, and Orphan Drug. The first UM171 cell therapy trial was launched in 2016, and we have now completed our phase 2 studies and treated over 115 patients across the US, Europe, and Canada with promising data, I'll show you in a few slides, which is, in our view, challenging the standard of care. UM171 cell therapy is manufactured through an efficient and reliable manufacturing process. Firstly, the stem cells are identified with a CD34 plus marker and are isolated. Then the stem cells are expanded ex vivo using UM171 and are harvested after a 7-day process in culture in very small volumes with a very small footprint. The resulting cells are cryopreserved and are shipped worldwide for simple thaw and infusion at bedside in the clinical centers. In terms of clinical results in stem cell transplant, GRFS, or GVHD, and relapse-free survival is a composite endpoint that has been developed to measure the overall success of an HSCT procedure. and takes into account the absence of non-relapse mortality, GVHD, and relapse. As you can see here, our clinical data suggests a higher GRFS than all other alternative grafts under the current standard of care, including compared to mobilized blood in a comparative registry study conducted by CIBMTR, and which has recently been published in Blood Advances. We have seen this data replicated in our phase 2 studies across different centers and in now over 115 patients. Therefore, we believe that this is indicative of UM171 cell therapy's potential to significantly improve long-term survival and patient outcome, and thereby challenging the standard of care. As you can see from this table, which is more directional in nature, UM171 cell therapy combines the benefits of each of the 3 different stem cell sources in a single best-in-class product. Stem cell transplants, or HSCTs, are a treatment for several blood cancers and diseases. For many patients, an HSCT, or bone marrow transplant, is the best and often the only curative therapeutic option. Our go-to-market strategy is based on a phased approach by first focusing on the high-risk patient segment and thereafter the broader bone marrow transplant, or HSCT, segment. Our adoption strategies has been and continues to be centered around close relationships with treating clinicians and bone marrow transplant physicians, of course, in key transplant centers in the US and Europe in order to drive awareness and adoption. So before the end of the current calendar year, we're excited about and looking forward to the following catalysts. So our end of phase 2 meeting with the FDA is booked and has been scheduled in November. We're also going to present at the ASH annual meeting of this year by our USPI Filippo Milano our phase 2 results from UM171 cell therapy, and 2 other abstracts will be presented on UM171, including one from Dr. David Williams at Boston Children's on the ability of UM171 to protect against DNA damage in a gene therapy setting. I see we're a bit short on time, so in terms of product improvement, I will only say that we are working on a 2nd generation product which promises to be even better. And now briefly about our preclinical pipeline. We've been optimizing our UM171 technology to expand and rejuvenate stem cells from mobilized blood and bone marrow. We've also developed an engineered HSC platform which leverages our cell expansion and rejuvenation technology. And finally, we've been developing novel and new small molecules in the field of cell and gene therapy. So as you've seen today, by making better cells, ExCellThera is focused on providing better lives for patients and their families. Again, my name is David Millette. I'm the company's CEO, and this concludes my presentation. Thank you.